start with one of the proteins. We tested many proteins, but two of these proteins turned out to be very interesting and intriguing. And one of the protein which we worked, and fortunately, Professor Y.P. Singh is here. He was the examiner of my student who presented that. He reviewed her thesis, and ultimately that work was published in Journal of Infectious Disease. So I wish that if I'm going to start with that work, but before I go to that work, I'm going to acknowledge first my students who ultimately worked with this, they contributed to this talk. So this is a team I had, a beautiful team. They worked with they cooperated with each other, helped each other. But the main work which I'm going to tell you today was done by Fatima and Sanpreet. Okay, Fatima mainly did work on ACR1 protein and Sanpreet on MPD64, and ably helped by the entire team. And this work was funded by MHRD, SPARC, SERB, and from CSIR. So let me tell you over here that macrophage, and this macrophage, the Mycobacterium tuberculosis, it resides over here. And in this red portion is Mycobacterium tuberculosis. So this tuberculosis bacteria lives in macrophages, and ultimately the molecules which are responsible to kill this bacteria, they are nitric oxide. These are free radicals of nitrogen or nitric oxide. These are oxygen intermediates, so through which the bacteria is killed. But the bacteria is so clever, it understands that what's going to kill me inside the cell, so ultimately it blocks it. So same way, the molecules which are responsible for its destruction, in case of dendritic cells, it blocks those molecules as well. See, to present antigen to T cells, so T cells get activated, and the T cells, they play a very cardinal role in destruction of Mycobacterium tuberculosis, and T cells, they are blind to see native antigen. So they can only see antigen when the antigen is complexed with the antigen or with the peptides of the pathogen. So now for presentation of antigen to T cells, MSC molecules, they're very important. So what Mycobacteria does, it downregulates the co-stimulated, downregulates MSC molecules so that the antigen is not presented. Right, so then the important, even though the antigen presenting cells, they present antigen in context of the Mycobacterium tuberculosis, but signal number 2, which is called as co-stimulatory molecules, they are quite crucial. If the first signal, which is MSC antigen complex, is presented effectively to T cell, but second signal, which is co-stimulatory molecules, if it is not presented, instead of getting T cell activated, the T cell, they undergo unresponsiveness. The T cell, they get tolerized. So here, what the bacteria does, this bacteria, it downregulates MSC molecules on dendritic cells, it downregulates co-stimulatory molecules on dendritic cells, right? So ultimately, the cells which are supposed to kill the bacteria, they get tolerized, right? So first, the bacteria realized that how to make the most potent antigen-presenting cell inefficient, that is dendritic cell. The bacteria knew first. Later on, immunologists, they identified that if first signal, second signal, the co-stimulatory signals are not delivered to T cells, they undergo NRG. And this is how, self-reactive, autoreactive T cells, they remain unresponsive in our body. Okay, so there are many mechanisms. So there are another mechanisms which the bacteria try to utilize. That is, the bacteria, when it resides and it multiplies inside the macrophage, so what it does, it induces necrosis in macrophages, and ultimately the macrophages, they burst, and now the bacteria is liberated, and these bacteria have now more opportunity to infect other macrophages, right? This is how they keep on infecting other cells of the body, and they spread. So these are some of the mechanisms, well-established mechanisms through which the Mycobacterium tuberculosis, it evades our immune system, right? So what we observe that the Mycobacterium tuberculosis, it downregulates even, toll-like receptors. So toll-like receptors, they are very crucial to activate macrophage or dendritic cells. And they can also downregulate it on T cells, which is important. So what we realize that if we can trigger the T cells, which have undergone, exhaustion, so the bacteria is so clever that you need optimum activation of T cells. What it does, it sometimes induces rapid activation of T cells. So they lose their energy and ultimately they go in exhaustion. So they are just useless. Something like, if you are a wrestler, first thing is what you do, your opponent, make him, run at towards you, make him keep on running. And once he gets exhausted, then usually you can beat. So it makes T cell exhausted, and what you observe that if you overcome this exhaustion of T cells, then these T cells can make bacteria. We also observe that it utilizes other pathways as well, like lipid sensing nuclear receptors, and through these receptors also, which are crucial for killing of mycobacteria, it can evade immune system by suppressing these molecules. And recently also we identified that it downregulates certain molecules. Again, these are innate receptors, and when these innate receptors are downregulated, the mycobacterium can survive. And if you can trigger through these innate receptors, then it induces autophagy. And through autophagy, then mycobacterium is killed. So while downregulating these receptors, it inhibits autophagy and then evades immune system. Right, so there are many mechanisms we were talking about, and then ultimately we identified one protein, which is ACR1 protein of Mycobacterium tuberculosis, is 16 kDa molecule, right? So here, what we observed that we took many proteins, at least 20 proteins of Mycobacterium tuberculosis, but this protein has unique ability to not to allow the cells to differentiate into dendritic cells. Right. In case if the bone marrow cells, which ultimately get differentiated into dendritic cell, while they're differentiating, they encounter ACR protein of Mycobacterium tuberculosis. So what happens? These dendritic cells, they undergo a state of tolerance, right? Or these dendritic cells, they MSC molecules are suppressed, the co-stimulatory molecules are suppressed, and the presence, the secretion of IL-12, which is important cytokine for differentiation of Th1 cells, they are also suppressed. So this protein is quite unique that it impairs the maturation of dendritic cells. And these dendritic cells, when their maturation is impaired, they lose their ability to activate T cells, right? So this was the work which was done by Fatima, and Dr. Y.P. Singh was the examiner. So here we showed that this ACR1 protein, it also decreases the decreases the phagocytic activity of dendritic cells. So in the upper portion, we took one protein. Okay, so this protein was we took antigen and we labeled antigen with FITC so that we can identify it through flow cytometry. Right, so when we looked into if you see upper portion, the histogram, these histograms, when dendritic cell encountered this protein, so it shows in a dose-dependent manner that the capacity of dendritic cell decreased to phagocytose this antigen. Okay. So this we showed through flow cytometry as well as through confocal microscopy, right? And then we observed that these dendritic cells, they have ability, they lose ability to stimulate T cells. So T cells, they lose their ability to be activated, and then these T cells fail to secrete interferon gamma. They fail to secrete, IL-2. Even they fail to secrete even Th2 type of cytokines. And importantly, that these dendritic cells, they lose their ability to phagocytose even Mycobacterium tuberculosis. So TB bacteria, what it does, it fires the ACR1 on bacteria, on the host immune system, and ultimately it attacks that cell of the immunity which is considered to be the cell, the antigen presenting cell that can only activate naive T cells. So naive dendritic cells, they cannot activate naive T cells, and ultimately they lose their ability to even phagocytose these bacteria. And importantly, now, instead of that, the macrophages or dendritic cell, they eat bacteria invades dendritic cell, and it survives there. So if you can see in this graph, over here, this bar diagram, it shows the left panel that when the DCs, they are normal DCs, controlled DCs, which have not encountered this antigen, they sufficiently kill the bacteria, right? So y-axis is Mycobacterium tuberculosis CFU, and the x-axis is towards the left is a normal dendritic cell, and towards right, these are dendritic cells that encounter this ACR1. So when they encounter ACR1, these dendritic cells, they lose the capability to kill mycobacteria. The mycobacteria which are inside the cell, this dendritic cell, it fails to kill it, right? So they lose the ability to phagocytose, and if the bacteria infects dendritic cell, then dendritic cell fails to kill this bacteria, and the bacteria proliferates inside these dendritic cells. Right, so then we deciphered what is the mechanism, and the mechanism was that it enhances all those molecules in the dendritic cell which are suppressive molecule, right? For example, these dendritic cells, they start secreting more TGF-beta, they start secreting more of IL-10, and they start producing more of IDO, right? So all those molecules which are responsible for suppressing the activity of dendritic cell, they are upregulated. They are secreted or expressed in high amount, whereas the molecules which are responsible for activation of dendritic cell, they are suppressed. And interestingly, when we did the phenotyping of the T cells, which is in the lower panel, this green flow cytometer dot plots. So here, what we observed that when we did phenotypic analysis of these T cells, majority of them, they were regulatory T cells, the cells which are suppressive, the cells which secrete TGF-beta, IL-10, right? And importantly, when we identified the molecules, the transcription factors which are responsible, we observed that it was working through STAT3. So STAT3 was ultimately upregulated, through which it's suppressed, and it is suppressed NF-kappa beta, NF-kappa B, and also it upregulated SOX3 and downregulated SOX2 and SOX1. So indicating that it upregulated all those transcription factors which are responsible for suppression and downregulated those molecules which are important for the activation of dendritic cells, right? So this was very important, that how this protein was utilized by Mycobacterium tuberculosis. And the side by side, we were also, we tested about 20 proteins, and 18 proteins didn't show any activity on dendritic cells. But then another protein which was important, this was MPD64. So what we observed when we tracked these cells, the dendritic cells, same way, while the bone marrow cells, they get transformed, they get differentiated into dendritic cells if they encounter MPD64, protein of Mycobacterium tuberculosis. So this is another protein. What happens to these dendritic cells? So the conditions which we provided, these were conditions in which dendritic cells, they are differentiated. That is, in presence of IL-4, and GM-CSF, right? So they become dendritic cells. But if you provide additionally, along with IL-4 GM-CSF, MPD64, now these dendritic cells, instead of becoming dendritic cell or their activity is impaired, rather they become MDSCs. Okay, they become myeloid suppressor cells, myeloid-derived suppressors. So this we observed by the fact that these cells, these cells, while they were being differentiated into dendritic cells, if they are not exposed to MPD64, they become normal dendritic cells. They activate T cells, right? They show high expression of co-stimulating molecule, high expression of MSC molecules, right? And they secrete all cytokines which are responsible for activation of T cells. But here, importantly, what we observe that these dendritic cells, which were differentiated in presence of MPD64, they express high level of PDL1. They express high level of TIM3. These both molecules, they're responsible for immunosuppression, right? So then we looked into the phenotype of these cells, and we observed that they express high level of Ly6C, which is a marker for MDSCs, myeloid-derived suppressor cells. But important part is that these cells, they didn't show any decrease in the expression of co-stimulating molecules like CD40, CD80, we measure CD11B, and of course there were down-regulation of MSC class 2, but co-stimulating molecules, they were upregulated. Okay, so instead of that, these cells, they become dendritic cells and show impairment in the activity. They were totally different cells and they become MDSCs. And what we observed that these MDSCs, they secreted the cytokines which were responsible for suppression of the immune system. So the middle bar, the black bar is of normal DCs. Then the middle bar is of DCs which encounter MPD64, and the right bar, or the gray bar, is a control protein of Mycobacterium tuberculosis, which we use CFP10, right? Or we also use ESET6. So the first A1 is IL-6, which got downregulated, IL-12 got downregulated, right? TNF alpha was downregulated. But IL-10, which is important for immunosuppression, it was highly upregulated, right? So again, it was a phenotype of dendritic cells, a phenotype of a suppressive cell. Now, when we did the SAM, scanning electron microscopy of these cells, so if you see towards the upper portion, the left first portion, this was a normal dendritic cell, and I don't know from whether you can see from there, it still have dendrites. So indicating that it was differentiated into a dendritic cell. But the middle one is which encounter MPD64 decreased in size and didn't show any dendrites on the surface. And the right side is the one which was incubated with CFP10. So CFP10 was more important. It was differentiating dendritic cells into dendritic cells. And these dendritic cells has they failed, they could not activate T cells. Okay, so here we have shown that they could not activate T cells, and in fact these dendritic cells, they had shown downregulation of CCR7, which is a chemokine factor, which is responsible for migratory activity of dendritic cells. So this was downregulated, indicating that such dendritic cells, they lose their ability to migrate. So let me tell the students that dendritic cells, what it does, if you have an infection over here, they're present in the skin as Langerhans cells. So these dendritic cells, they're going to migrate to the site of infection, they're going to capture antigen, and then they go to lymph nodes. So the migratory activity is very important. So if it is impaired, they cannot take antigen to lymph nodes. There, they prime T cells. And when T cells get primed, they proliferate over there, they expand, and ultimately they reach the site of infection. This is how they control infection. So what we observed that there was a downregulation of CCR7 and other chemokines as well. And if you see towards right, what we did, we sorted these cells, which were expressing, chemokine 7 receptor and cultured with MPD64, showed downregulation. So what we did, we injected in animals, and what we saw was that such dendritic cells which were incubated with MPD64, we could not see their population in lymph nodes. or in your spleen, they were more aggregated at the site of injection, right? So ultimately, such dendritic cells, they lost their ability to migrate. So this was very important observation that dendritic cell function is not only impaired, rather they lose their ability to even migrate. Okay, so this also we checked through Nile red, which ultimately indicates its uptake, indicates the suppression activity of dendritic cells as well. And finally, what we did, we also did the activation of T cells, both CD4 and CD8. The dendritic cells have capability, they can cross-present antigen to CD8 cells. So not only CD4, they activate, even they can activate CD8 cells. So here, again, if you see the black bar, this is a normal dendritic cells activating CD4 cells, and the middle bar, which is a dendritic cell incubated in presence of MPT64, and then dendritic cell, the gray bar, they are cultivated in the presence of CFP10 or ESAT6, and towards the right, extreme right, is a T cell, which is a positive control, is stimulated with anti-CD3 28. So the middle one, just next to black, you see that CD4 T cell, they could not proliferate, CD8 T cells, they could not proliferate, and we did in both cases. These are antigen-specific proliferation as well as allogenic proliferation. So in both cases, we could not see proliferation. There was a down-regulated proliferation of T cells, CD8, CD4 cells, as well as there was a decreased secretion of interferon gamma, IL-17 was decreased, but somehow, if you see this bar diagram, in this case, IL-10 was increased, TGF-beta was increased. So suppressive cytokines, they were increased, whether it was the cells, they were activated in antigen-dependent fashion, or they were activated through allogenic reaction. So in all cases, Th1 cells, Th17 cells, they were suppressed, and Tregs, they were increased. So what we did, that we injected these animals with MPT64, and just to check whether what happens in vivo condition. And when we monitored CD4 cells phenotyping, we did phenotyping of CD4 T cells. So it was very interesting to note that the majority of the cells, they were expressing FOXP3. That is, they were regulatory T cells, the cells which are important for suppressing immune system. Right, so then what we did, we wanted to see whether these MDSCs, do they retain the power of phagocytosis? That is, can they eat bacteria? Can they phagocytose bacteria? So these MPT64 exposed dendritic cells, they lost their ability to phagocytose bacteria, and we observed this whether we took H37RA or H37RV, non-pathogenic or pathogenic strain of the bacteria we took, or we took non-pathogenic Mycobacterium spagmatis, and we also took dextrin FITC as an antigen. In all four cases, we observed that the phagocytosic activity of dendritic cell is impaired. Okay, so this we did by flow cytometry, we did by CFU assay, and we also showed by confocal microscopy that the phagocytosis ability of dendritic cell is impaired upon exposure to MPT64. And same way as I told about ACR1, over here we also observed that the bacteria, they could proliferate inside the MDSCs, which were exposed to MPT64. So if you see the black one, right, so y-axis is the CFU or Mycobacterium tuberculosis, H37RV. The black bar indicates normal disease. So these normal diseases had killed majority of the bacteria, but if you see gray bar, these MDSCs which encountered MPT64, they had impaired ability, or they have lost their ability to kill Mycobacterium tuberculosis. And ultimately, we deciphered the mechanism, how it's working. We observed that the transcription factors which were responsible for that, these were involved in it. So mainly those factors which are suppressive in nature, they were upregulated, and were responsible to activation of dendritic cells, they were suppressed. For example, arginase, which is a suppressive factor for dendritic cell, it was highly upregulated. Okay, and we showed arginase by biochemical method and also by Western blot. And we also saw that STAT3, it has increased, which is ultimately, it induces a transcription factor of inducing suppression of dendritic cells. IDO was increased, IDO is also a suppressive factor. And we observed that NF-kappa was drastically decreased. Okay, so indicating that it is working through these molecules inducing suppression. And these dendritic cells, what we observe that they didn't have capacity to uptake glucose, right? And they were very rich in fatty molecules. They were rich in fatty acids, and they had impaired ability to uptake glucose, indicating that these dendritic cells, they have lost their ability, metabolic activity as well. And then we also looked into that whether these dendritic cells, they have ability to take methyl glycol. So it was observed that these dendritic cells, or the MDSCs, they showed more of methyl glycol in them. Right, and this we showed through flow cytometer as well as through confocal microscopy. So you can see here that these red granule or the red ring, it shows the high presence of methyl glycol in these dendritic cells. Plus, we also observed some important immunosuppressive molecules, and like glutar1 and HY2 and GLO2. So these molecules, S440, we observe these are all suppressive. These are, GLUT2 and HY2. These are immunosuppressive immunoactivators, so they got suppressed, and the molecules which were responsible for activation, they were suppressed, and responsible for suppression, they got activated. And finally, we observed that the inhibition in the proliferation of T cells, whether it was CD4 cells or CD8 cells, this was because of high secretion of nitric oxide. Right, so usually it is known that nitric oxide is responsible for bactericidal activity. But on T cell, it's been discovered recently that the nitric oxide can suppress the activity of T cells. So here what we observe that when there was a high, in case of cultures, we observed that there was a high secretion of nitric oxide, and more than nitric oxide, and more was the suppression in the proliferation of T cells. So finally, it appeared that the inhibition in the proliferation of these T cells, whether these are antigen specific or we showed in case of allogenic, this was because of high secretion of nitric oxide. So if you see in the down panel, the bottom panel, this is nitric oxide, and then Western blot we have shown that iNOS is also decreased, and which is responsible for transcription factor. STAT1 is responsible for this. So this is the mechanism which is operating is through nitric oxide, and we have shown that is iNOS and STAT1 transcription factor is also decreased. So in conclusion, that Mycobacterium tuberculosis is a very clever bacteria, right? And it understands how to block. It secretes this antigen. It's not only the antigens which is to stimulate immune system, whereas the immune system try to utilize Mycobacterium for its benefit. But at the same time, Mycobacterium also utilizes the host for its benefit as well.